In this second video on the central dogma of biology, we're going to look at transcription, the first step in gene expression. Gene expression refers to the idea of a cell accessing the information in its genome. Remember, the genes are essentially recipes for how to make a protein. And so transcription is the first step in accessing one of those recipes so that in the second step, translation, the actual protein can be properly built. So let's look at the basics of how transcription takes place. So here's gene expression in kind of a summary. On top, you've got double-stranded DNA. You can see the base pairing. You can see the reverse orientation to one another. Transcription makes a single-stranded RNA copy of what was a double-stranded DNA gene. Okay, so transcription makes a single-stranded RNA copy of what was a double-stranded DNA gene. We call that single-stranded RNA copy of a gene a messenger RNA or an mRNA. In the final video that I'll show you after this, we're going to see that ribosomes latch onto that mRNA, and with the help of a variety of other proteins and molecules, that mRNA gets translated from the language of nucleotides into the language of amino acids, and a protein is produced. So let's look at that transcription step here. In transcription, the first thing I want you to notice is something called transcription initiation. A gene is not somehow physically determined to start here and stop here. It's informationally determined. In other words, there's a sequence of A's and T's and C's and G's that says start here. And that sequence we call a promoter. And that's what we're showing here on the left here in yellow on the top strand that we've got, this top molecule. And then if you move over to the right, you see another sequence that we call the terminator. The terminator says stop here. So our gene is going to extend from our promoter through what we call our coding sequence to our terminator. Now what's going to happen to get this going is something called initiation. Initiation is when RNA polymerase, an enzyme that polymerizes an RNA molecule, is guided to a promoter with the help of a protein called sigma factor. Sigma factor is what's known as a transcription factor, and there are a variety of these in different living organisms that guide their RNA polymerases to the appropriate promoters. During transcription initiation, RNA polymerase and sigma factor bind to the promoter, and they create this initiation complex. In the second step, RNA polymerase can open up a small bubble. We'd call this a transcription bubble, similar to the DNA replication bubble that we saw in DNA replication video. A small opening where we've got some single-stranded DNA, and it's going to move from the promoter towards the terminator. And as it moves from our left to our right, it's going to peel apart these two strands. It's going to read one of the two strands, use it as a template, and add the appropriate base pair to that template while linking those RNA nucleotides together. Now, as the ribosome, the RNA polymerase moves past a region, the two strands of DNA will actually collapse back onto themselves and base pair, and our RNA molecule that's growing will be sort of dangling out in the breeze. So that's the initiation of transcription. Elongation is this process of moving from our right to our left. In this diagram, you can see the red molecule that represents our RNA molecule, and you see how the only place that it's actually base pairing with one of the two strands of DNA is right in that bubble, right where the RNA polymerase is sitting. Where the RNA polymerase has already moved from left to right, the two DNA strands have closed back up again. In other words, they have come back together in their base pairing patterns, and the RNA is just sort of dangling out away from the RNA polymerase. In the little inset, you can see the purple template DNA strand, and you can see these individual RNA nucleotides that come in as nucleotide triphosphates, just like ATP. They're in a high energy state, so they provide the energy to actually form the covalent bonds with the growing RNA strand. But you notice they're following the base pairing rules. And don't forget that in RNA we don't have thymine, we have uracil. And in DNA we don't have uracil, we have thymine. So uracil on the RNA molecule will bind with adenine on our DNA molecule. So keep that in mind as one of the differences during transcription. So this is elongation of the RNA transcript. And then finally, we have termination. When the RNA polymerase reaches the terminator sequence, there are some steps involved that allow the RNA polymerase to let go of the DNA and let go of the growing RNA transcript. We call this a messenger RNA transcript, and it is now almost ready to be read by our ribosomes in the next step, which is translation. In bacteria, this is ready to go. There's no processing, no modifications to that messenger RNA that needs to take place. However, in eukaryotes, it's a little more complicated. So you see the diagram of a gene on top. Eukaryotes have something called exons and introns. Think of the introns as sequences of DNA that are intervening between exons, and the exons are the sequences that will ultimately be expressed. That's one way to think about it. So we see that we've got a promoter, start of transcription, and somewhere beyond exon number 3, we're going to have a terminator. When transcription first takes place, we create what's called a primary transcript. The messenger RNA will have all the exons and all the introns in place. But before that can be translated, the introns have to be snipped and cut out very, very precisely, and the exons have to precisely be covalently bound back together. So you see the mature transcript in a eukaryote down at the bottom, represented by the blue, yellow, and red boxes. That's an mRNA that only has the exon sequences, and the intron sequences have been completely removed. 2 other steps happen. Something called a cap is added at the left-hand side of this transcript, and a tail is added at the right-hand side of this transcript. And all that is required for this molecule to be exported from the eukaryotic nucleus and then attached by a ribosome for translation to take place. So that's all I have for you for transcription. So in the final of these 3 videos, we're going to look at the actual translation process itself.